The first step is gene expression. What I'm going to do is I want to see when I have the information, I'm transcribing. So first step of gene expression, DNA has the ultimate code, so transcription. I'm taking DNA and I am making mRNA from it. So this is my DNA. I've taken the supercoils and everything off of it just to make it simpler. This is my gene. Every gene starts with a beginning called promoter. And by the way, for this, there's regulation that the chapter talks about. Regulation of gene expression, I don't care for you guys to know. You're not cellular biology, you're never going to use this. So this portion that starts is a specific sequence. That sequence is referred to as the promoter. It promotes the gene, like a promoter for an artist. When that gene needs to be expressed, promoter is active. It says, come transcribe me. Come here, you need me. And every gene has to say, you start here, that's promoter, you end here, we are done, no more information. That one is called the terminator. Okay? Imagine you have to get past Arnold Schwarzenegger, so you can. All right? Terminates, promotes. Promotes starts. Remember, DNA is double-stranded. So one strand is 3 to 5, the other strand is 5 to 3. There is only one enzyme in this whole process, and this enzyme does it all. The name of your enzyme is RNA polymerase. That's it. The whole process of transcription, RNA polymerase does it all. Doesn't need anyone. When promoter is active, your RNA polymerase goes and attaches to the promoter region. If promoter is not active, RNA polymerases cannot attach. Promoter becomes active. Don't care for you guys to know how. Magic. Active. RNA polymerase attaches to it. Then RNA polymerase does everything. Remember, the strands of DNA have to be separated so you read the nucleotides. RNA polymerase unzips, moves from promoter to terminator. Always has to go from promoter to terminator sequence. Your mRNA is single-stranded, so it doesn't have to read both strands. It only needs, it chooses one strand to read. So as it unzips, finds a template, reads the template, and makes the mRNA complementary to that template. The template actually has a name. We call it the sense. It makes sense. It's the template that mRNA is made based upon it. Rules of DNA is what? How do you read DNA? 3 to 5. Don't say right to left, say 3 to 5, because depending on how I labeled this, it could be different. So 3 to 5. RNA polymerase is not insane. It already has way too much to do. So when it starts doing this, it says, all right, I have to go from P to T, promoter to terminator. I have to unzip and separate. And then at the same time, I have to read. So guess which one of the templates, which one of the strands is it going to use? In my example, the top one or the bottom one? Top one. It's not insane. It's not going to go here and then back its way. It will choose the strand that is going from promoter to terminator in the direction of 3' to 5'. So it will unzip, and we call that, so when it attaches is initiation. Then it's elongation and termination. I don't even care for you guys if you know those terms. Just know, promoter is going to attach, unzip, create this little bubble frame, breathing frame for itself. Then read that, and as it moves, so let's say it has gone here now, so the RNA polymerase, as it goes there, reads that strand and makes a complementary 5' to 3' mRNA. It does this all the way until it gets to terminator. When it gets to the terminator, because of the sequence of the terminator, it knows that it has to fall off. RNA polymerase falls off, releases the mRNA, you're done with transcription. Very simple. Again, promoter. Every gene starts with a promoter, ends with a terminator. The enzyme that is responsible for transcribing is called RNA polymerase. RNA polymerase attaches to an active promoter, unzips, reads, and makes mRNA. Unzips going from prime promoter to terminator. Reads the template that goes 3' to 5', going from P to T, makes a complementary mRNA. That's it. They're done with the process of transcription. Let me show you the animation for it, and then I'll promise I'll let you go. So again, I have this. So here, again, the top one is 5 to 3. The process is actually not just one RNA polymerase attaches and goes. As long as the promoter is active, the second the promoter is empty, another RNA polymerase. The big mistake students make, they start adding helicase, ligase, and all of that to this process. No, it's even better than that. It's just an RNA polymerase promoter, terminator, that's it. You know the process. So keep it simple. Again, I have 2 animations for you guys.
So let's start off with having a look at transcription. Overall, what happens in transcription is that a cell makes an RNA copy of DNA. The cell shown here shows that transcription occurs in the nucleus of the eukaryotic cell. In this figure, DNA is highlighted in blue and RNA is highlighted as green. Transcription is broken down into 3 stages: initiation, elongation, and termination. Let's have a look at the first stage of transcription. Initiation is the first stage of transcription, and it's during this time that enzymes unwind the DNA double helix, and a specific enzyme called an RNA polymerase attaches to a region of the DNA called the promoter. Now, this promoter region of the DNA actually signals the start of a gene. Following initiation, elongation is the second step in transcription. So once the RNA polymerase is attached to the DNA of the promoter region, it moves along the template strand of DNA, and as it moves along, it adds nucleotides one at a time to extend the RNA strand. The final stage of transcription is termination. In termination, the RNA polymerase reaches a region of DNA referred to as the terminator sequence. When it reaches this region, it signals the RNA polymerase to stop adding nucleotides to the RNA strand and to separate from that DNA. The RNA strand is then released from the RNA polymerase, and this ends transcription.
Alright guys. So, thanks for putting up with that. I have your tests. If whoever wants to see it, look at it, stay. If you don't, free to go. Not forcing anyone. So this weekend, your mission, should you choose to accept it, is to go through RNA replication. Once you have that, if you want, start with transcription. But other than that, just do replication. If you don't get to this, just be familiar again, what is RNA?